see lots of faces popping up on the screen. It's so good to see you. Welcome to our Lunch with Docs today as we discuss whether PD is a single disease or not. Before we get started, just a bit of housekeeping. First of all, my name is Anissa Mitchell. I'm with PMD Alliance, so I'm glad that you're joining us. And I would love it if you would pop open your chat box and give us a shout out and let us know where you're joining us from. Dr. Mari and I were just talking a few minutes ago about the beauty of Zoom and how we can join with one another across the United States and even outside of the United States. So please let us know where you are joining us from. And that is also where you're going to ask your questions today. So I'm sure there will be some as we dive into this topic. It's a very exciting topic. So feel free to ask your questions there, and we're going to hold those until the end, but we will go back to those. So I'll be collecting those for Dr. Mari. And I know he has a lot of material that he's going to share with you. So I'm going to introduce him and then I'm going to push it over to him. So for those of you who have not had the pleasure of hearing Dr. Mari speak, Zoltan Mari is the Ruvo Family Chair and Director of the Parkinson's Disease and Movement Disorders Program at Cleveland Clinic Lou Ruvo Center for Brain Health in Las Vegas, Nevada, and also Adjunct Associate Professor of Neurology at Johns Hopkins University. We're very excited to have him speaking today on whether PD is a singular disease or not. And so I am going to welcome Dr. Mari, and I'm going to hand this over to you.
Thank you so much, Anissa. It's my pleasure to be here again. I really love to talk to this wonderful group, and we always have good conversations and questions. And as mentioned, please don't hesitate to post your questions. We will try to get to as many of them as possible. And with that, without further ado, I would like to share my my screen and get started. And while the material and the topic is indeed very voluminous and substantial, I don't have a whole lot of slides, and it's purposefully so, because I don't want to present busy slides and we bog down reading them. I would like to highlight a few interesting related issues, and hopefully we can expand on them, and maybe with your interactions and questions focus it further. We sort of did that a little bit with Anissa earlier with her related questions, and I think that that is a good format. And of course the topic is very interesting to me, and probably to most of us. I do get this question in clinic from other patients, but certainly in conversations with colleagues, whether we can consider Parkinson's disease a single disease or not. And the or not, the alternative to a single disease would be a syndrome. And some of you may not quite understand or know exactly what a syndrome versus a disease, what is the difference. And many times in clinic, I do discuss that question in the context of Parkinson's disease, assuming that that is a single disease and a single so-called clinical pathological entity, versus Parkinsonism, which is by definition a syndrome. So there's no question about that. And so I would like to start with that explanation or expansion on what is the difference between Parkinson disease and Parkinsonism, and that could give you a bit of an intro as to what we think about a disease versus a syndrome. So a disease is a well-defined clinical pathological entity. What that means is that there are certain clinical diagnostic criteria that are very strict and rigid, and they are very well defined and described. And so when somebody meets those clinical diagnostic criteria, we go through a list of check boxes to make sure that certain signs and clinical findings or exam findings are present versus absent. And so when we say that the diagnosis of Parkinson's disease is a clinical diagnosis, we mean that we apply this list of various criteria in a strict manner, and somebody either meets those criteria or they don't meet those criteria. And so the clinical pathological entity, the second part, the pathological, is where we actually find certain pathological features that can only be accessed through the examination of brain tissue, for instance, and it may include immunohistochemistry, very specific pathological methods and stains that will highlight certain pathological features under the microscope usually. And in Parkinson's disease, I will show you some of those slides that we can see under the microscope, and they say that the Parkinson diagnosis is clinical, but the definitive diagnosis is pathological, and I will get to that. Now, what about Parkinsonism? Parkinsonism, by definition, is a syndrome, which is basically a constellation of certain symptoms. When certain symptoms and findings are seen together, then we call that a syndrome. And in the case of Parkinson's, the constellation of those findings include tremor, as well as slowness of movement or bradykinesia, and rigidity, increased muscle tone, and postural abnormalities. Now, when these symptoms are seen together, that's Parkinsonism. If somebody has Parkinson's disease, then they certainly will have those symptoms. So in other words, everyone with Parkinson's disease meeting the clinical diagnostic criteria that I mentioned earlier will also have Parkinsonism, because Parkinsonism, the way that these symptoms look like, is a manifestation, is a sign of Parkinson's disease. The thing is, though, that it is not only Parkinson's disease that can produce those signs and symptoms. So while every patient with Parkinson's disease will have Parkinsonism, the appearance of these Parkinsonian signs, not everyone who has those signs or the appearance of those symptoms will actually have Parkinson's disease, because there are other conditions that can produce very similar signs. And those other conditions that produce Parkinsonism, but they are not Parkinson's disease, those other conditions include so-called primary and secondary forms of Parkinsonism. So I don't want to confuse, go ahead.
I'm sorry, I don't want to interrupt you, but I don't know if you intend to be on a different slide than the initial slide.
I can change that.
I didn't know if you were actually on a different slide than your intro slide, because I think when we did this before, there was an issue with the initial slides moving.
No, I was just talking too much.
Okay, all right. I just wanted to make sure, because I didn't want to miss anything.
I meant that discussion as sort of an intro as to what is disease versus syndrome. And these are just my disclosures. And this is my outline, which basically I spent the initial few minutes on basically discussing what is a disease versus a syndrome. And again, the difference is the syndrome is always a broader category. Syndrome is when there are certain signs and symptoms occur together. That is a syndrome. Now, it doesn't say anything about the cause of those signs and symptoms. It only says that those signs and symptoms are seen together in the same patient at the same time. So coming to Parkinson's, the Parkinsonian syndrome, or in other words, Parkinsonism, is a syndrome, and it includes tremor and bradykinesia or slowness and the other motor symptoms that I mentioned, and then it's a syndrome. It does not say anything about the cause, why somebody has those signs and symptoms. And turns out that it's more than just one cause. So as far as Parkinsonism or the Parkinsonian syndrome, that's a classic syndrome with multiple different causes. So that's not a single disease. However, for the longest time, we all taught, and we were taught, that Parkinson's disease, on the other hand, is a single disease entity, a single so-called clinical pathological entity where there is a single cause, a single pathology, and a single look and feel. And well, the topic of today's talk is mostly to say that that's not the case, or probably not true. In other words, Parkinson's disease, to further complicate matters, Parkinson's disease itself seems to be a syndrome too. And Parkinson's disease, even the same pathology that we know, and I'll show you in the next slide, can in fact come from more than one possible causes. And so that is what we would like to discuss here. And that's an important question, because there are certainly major practical ramifications of the differences between individuals who all have clinical diagnosis of Parkinson's disease. They all meet those criteria, so they have Parkinson's disease, but still, because it's not a single disease, because there are multiple possible causes, it is very important to learn more about that, because the treatment could be different. The prognosis could be different. And those differences in the subtypes and the differences in the combination of causes could lead to very systematic and consistent differences in prognosis, or how we should treat these diseases. And I think it is becoming more and more relevant. Now, when we think about the last 50 years of Parkinson treatments, we think that the dopaminergic system and how treatments that were developed to fix the problems with the dopaminergic systems work for everyone. There is no need to really, consider or think too much about how different patients are from one another when levodopa works for all of them. There was not much of a strong need to differentiate that way. So from this perspective of pathology, the clinical manifestations, and the treatment, the mainstream treatments, Parkinson's disease was one. So what's different today? Well, first of all, we learned a whole lot more about the causes of Parkinson's disease at a molecular level. We learned about genetic forms of Parkinson's. So we have built a knowledge base anchored in molecular knowledge of the causes and the underlying pathology, which is not the same across patients. But more importantly, we are trying to develop the next level of treatments, which are so-called disease-modifying or neuroprotective treatments. And those are distinctly different from one person to another in terms of what the target should be. And while many of these different causes and types of Parkinson's pathologies and etiologies will converge to something that is connected or similar or uniform across patients in the destruction of the nigrostriatal dopaminergic system and making them good candidates for dopamine replacement therapy. While that's still true, when we think about how to prevent, slow, intervene the initial strike and the initial molecular problems that started the disease in the first place, and may be different among individuals, when we want to target there, then we definitely should be thinking about the differences and the subtypes, and not think about what's common among Parkinson patient individuals, but what is different between them, because understanding that and specifically targeting different individuals with different treatments with that knowledge will be the key to be able to prevent, slow the progression of Parkinson's disease. And now I'm going to the next slide. I have very few slides for why I may linger on a single slide for relatively long, because normally when I give a talk, the rule of thumb is 1 minute per slide, and that's certainly not a rule of thumb applicable today. So I talked a little bit about the pathology and the definition of Parkinson's disease by pathology, and how the so-called definitive diagnosis of Parkinson's disease is made based on pathology. I just wanted to show you a transection of the midbrain below at the bottom part of this figure, and you see this little blackish or darkish stripe or line on the left figure, and that is blackish or this colored because of the pigment in those neurons. And the substantia nigra has these granules on the right hand side. You can see that the cytoplasm is full of these little brown pigment granules. And so on the bottom side, on the bottom of the slide, there is another transection from a Parkinson patient in the midbrain where it's pale. You don't see that colored stripe, and that's because those neurons that have the neuromelanin are dying and were reduced in number. Now, these are the neurons that are very critical to Parkinson's disease motor symptoms, the so-called nigrostriatal dopaminergic neurons. These are the ones that have those pigments for what gives the substantia nigra its name. Nigra means black in Latin. And so this is basically how the pathology and the definitive diagnosis based on pathological criteria has been set. Now you can also see these pinkish, much bigger bodies inside those cells, and those are the Lewy bodies, which is one of the most important pathological hallmark of Parkinson's disease. Now, this is something that I'm showing you again necessary for the definitive pathological diagnosis of Parkinson's disease that connects patients. If you look at just this, then you could say that Parkinson's disease is a single disease. In fact, if somebody doesn't have these signs and pathological findings, then they don't have Parkinson's disease. So from that standpoint, you could make an argument that yes, Parkinson's disease is a single disease as defined by those Lewy bodies and other pathological hallmarks, as well as the clinical diagnostic criteria. This is just a larger magnification, and you can see the cell in the center of this histological slide in a more blown up larger magnification, and the little halo around this hyalinaceous body inside the cell is the Lewy body. And then you can still see in the upper part of the cell those neuropigment or neuromelanin granules. So just to put that into perspective, I showed you these transections earlier, but where in the brain is the midbrain? And so this is showing it in the larger picture in the whole brain. You can see the individual here. This is where the brain is, and the midbrain is part of the brainstem, and this bluish structure here it represents the location of the substantia nigra, and those neurons that I was showing you in the earlier slide are the ones that will project these long axons into the so-called striatum, which is known as caudate nucleus and putamen. Those two together will make up the striatum. That's where those neurons project, and that's where the dopamine is released. And so this is a cartoon that shows the dopamine release. So again, this transaction takes place in the striatum, and normally those dopamine cells with those terminals up in the striatum will release the dopamine at a very constant rate, or more or less constant rate, creating a fairly steady dopamine level in the synaptic cleft, and that's the normal state. In Parkinson's disease, with the decline in the dopaminergic cell numbers, there will be less production and release of dopamine in those synaptic terminals, and that leads to a lot of problems down the line. Number one, those dopamine receptors, which are the structures that are meant to capture the dopamine and turn it into a nervous signal. This is how the neurons chat with one another. They will be upregulating, and that will lead to a lot of problems such as dyskinesias and motor fluctuations and many other late complications. So with that, I would like to go back to the question of what is it that makes Parkinson's disease a single disease? And what is it that it makes it not a single disease? And first of all, my personal opinion, and increasingly more and more experts would agree that Parkinson's disease should not be handled or treated anymore as a single disease. There are many practical ways of treating a syndrome. And many times the treatment is the same or similar, and therefore maintaining a single entity in a clinical world can have significant practical benefits. For instance, if somebody has imbalance, that's not even a syndrome, it's just a symptom. There could be a number of reasons why somebody has imbalance, a lack of balance, lack of postural reflexes, and tendency to fall. It could be from a number of diseases. It could be multifactorial. Yet, treatments are oftentimes the same. Balance therapy, for instance. So we don't necessarily just treat a cause. We can treat the end result in the form of so-called symptomatic treatments. And from that standpoint, I'm not arguing against dropping Parkinson's disease as a clinical entity, which is relevant in the clinical management sphere and domain, because many treatments will remain the same. If you have certain symptoms and symptomatic treatments are available, it almost doesn't matter where you come from, what was the original cause that led to that symptom, if a treatment treats it anyway. There are many other examples. Tremor is also somewhat in that category, not for all tremor treatments, but some of the tremor treatments, including occupational therapy, specifically targeting the occupational impact of tremor and the use of heavy utensils or utensils with the gyroscope and various other devices. They will help tremor no matter what the cause of tremor is, and there are many causes of tremor. So when I say that Parkinson's disease is not a single disease, I mean that it's not a single disease for the heterogeneity, the various causes of that can lead to Parkinson's disease. And in that regard, it is not a single disease, because we know that there are many different mutations that can increase the risk of Parkinson's disease. And these are called risk genes. And there are several. It's not just one. And we haven't even fully explored how a multigenic risk factor can result from a constellation of more than just one gene. But even if we talk about the single gene, the monogenic forms of Parkinson's risks, there are many of those as well. Now, when I say risk gene, it's important to realize that even if you have a certain genetic predisposition or carry the mutation that increases the risk of Parkinson's disease, it is never a certainty. It is never going to be an absolute certainty and just a question of time to have Parkinson's disease. Most of those risk genes will elevate the risk of Parkinson's disease, meaning you will, all things being equal, you will have a greater risk of coming down with Parkinson's disease compared to control individuals who don't carry that mutation. That's the definition of a risk gene. We know many of those already, and more are being discovered. Now, it is very interesting to also add that the clinical manifestations, the way that patients look and how their tremor or non-motor symptoms evolve over what period of time, to what severity, that also varies with some of these genetic forms. And the most important example is the Parkin gene. If somebody has a Parkin gene related form of Parkinson's, it's so distinctly different from the sporadic idiopathic form that some experts started calling it Parkinism rather than Parkinson's disease. Similarly, we know that many environmental factors, toxins, poisons, pesticides, herbicides increase the risk of Parkinson's. And it has been known that some of those, like the MPTP related form of Parkinsonism, is quite specific and different from the idiopathic form. Now, again, from this heterogeneity in terms of manifestations, as well as prognosis, we can build a much better and much more granular classification and subtyping of Parkinson's disease that can help us recommend with regards to treatment, but also advise patients with regards to prognosis, because we are finding that the various forms of these Parkinson's genes and etiologies and subtypes of Parkinson's can actually behave differently over time. One very interesting aspect of that is the non-motor manifestations of Parkinson's disease: cognitive impairment, depression, Parkinson's psychosis, anxiety, panic attacks, etc. The neuropsychiatric aspects can be very different in these different subtypes. And of course, that very strongly speaks to prognosis as well, and just planning for the future. Even if we don't currently have specific treatments to slow or stop the progression in these various subtypes, or we can still prepare patients better, or the families better, as to what to expect in the future. And actually, there's a lot of very interesting research going on currently in trying to improve our abilities to prognosticate. Now, when we talk about how we prognosticate, how we tell patients the way we think they will look in a year or 5 years from now, will relate to progression markers and prognostic markers, subtyping, and a much better understanding of their disease. However, we also need to recognize, and perhaps less research has been committed in this particular question of heterogeneity among Parkinson patient individuals, is that Parkinson patients also have other conditions. They have also other genes that have nothing to do with their Parkinson's. They have diabetes, or they may not have diabetes. They may have neuropathy. They may have cerebrovascular disease. They could have many important so-called comorbidities, other health issues that can modify and alter the way that their Parkinson symptoms and disabilities will fare over time. There has been very little research into this, but it is huge. A patient who has very significant degenerative joint disease and neuropathy, and they are in the wheelchair anyway, will be robbed of the ability to make their motor system work better, because they can't move from another condition, and that seems to accelerate their progression in a way that is not really the Parkinson disease. It's a deconditioning effect, but there could be actual specific pathological correlations. Very little is known about other neurodegenerative conditions and pathologies going on in Parkinson's disease. We know that Alzheimer's disease is far more prevalent in patients who have Parkinson's disease. These are so-called proteinopathies, where the underlying pathology greatly relates to a protein, a particular molecule that we all have in our brains, misbehaving and misfolding and becoming toxic. And it turns out that the various forms of these so-called proteinopathies interact with one another. The main protein, besides the amyloid in Alzheimer's disease, is a protein called tau, and we oftentimes classify Alzheimer's as a tauopathy because there's something wrong with the protein tau. On the other hand, Parkinson's disease is oftentimes called a synucleinopathy because the key protein that is playing a key role in the neurodegeneration, the process that kills the brain cells, the neurons, is the alpha-synuclein protein. We all have alpha-synuclein. It is part of the normal brain, but when it misbehaves, it starts spreading the pathology within the brain, killing cells. Now, these two different proteinopathies, the synucleinopathy, which we have in Parkinson's disease, and then tauopathy, which we have in Alzheimer's and many other diseases, if one is present, the other one is more likely to start. And then not only that it's more likely to start, but it's also more likely to spread a little bit faster and maybe more toxic. And so those interactions are also changing, even if somebody has the same genes and the same environmental exposures. Even if that's the case, the truth is that it could be very different because of the presence of another process that will alter the ongoing process, and that too will make Parkinson's disease not a single disease, because if we come down to devising a unique treatment plan to this patient, then it would be necessary to take those differences into account and develop an individualized, customized treatment plan that is specifically targeting the main problems that that individual patient is having. Now, that takes us to the so-called precision medicine. And you may or may not have heard this term before, but precision medicine is a buzzword that you will certainly hear more and more. Precision medicine is essentially what I tried to describe just now, which is that rather than treating diseases, we are treating individuals. Now, in many ways, we have already implemented a related approach when it comes to symptomatic treatments. Symptomatic treatments are not targeting the underlying causes of a disease, they are targeting the results, the symptoms of a disease. And these include physical therapy, occupational therapy, pharmacotherapy with symptomatic treatment agents, and many other things. And that approach is referred to as integrated care or multidisciplinary care, where somebody will treat the anxiety, an expert in anxiety, including medications, including mental health, counseling or therapy. Somebody will address the balance problems with the use of occupational therapy and balance therapy and physical therapy techniques. Somebody will prescribe the medications according to the symptoms of the patient. So that is already happening, especially in the settings of centers of excellence. On the other hand, we do almost absolutely nothing that is customized and uses this precision medicine approach when it comes to the causes and the genes and then the individually different targets and underlying disease mechanistics in Parkinson's disease or pretty much in many other disorders, where such approach described with precision medicine is taking a little bit better shape or a little bit more ahead in the game is cancer, where they can use very high techniques, very high advanced technologies to to type the cancer at a molecular level and develop antibodies or other treatments that are very specific to that particular cancer and that particular cancer cell by typing their membrane receptors, etc. We are not there with Parkinson's disease, but we hope to get there where we could just come to any individual with Parkinson's disease at any given point in their Parkinson's disease and develop that highly specific typing of their Parkinson's disease and offer treatments that are specifically meant for their that individual only. And I think there was a somewhat interesting James Bond movie recently, I think it was last year, No Time to Die, in which one of the ideas was that every individual can be genotyped. Genotyping is essentially to analyze all the genetic material that a person has. And I would add that we could also one day in the future analyze all the receptors and receptor subtypes and enzymes, which are not just the genes. So when we think about the genes determining features, we have to also add that even two identical twins who share their genetic materials perfectly will not be the same, because some of the genes can be modified after birth, and certainly after they started developing as embryos. But even after that, there are environmental interactions that will affect the manifestations of those genes, which can vary from one individual to the other. There is a word to describe that called epigenetics. And so the bottom line is that in the future, it may be possible to characterize an individual's entire DNA, as well as what the DNA did to them. So their bodily features, their receptors, their enzymes, their subtypes of their enzymes, etc. And once we have that enormous database describing this person perfectly, then we would know what specific treatment could be developed that work for them best. In the movie, it was used to kill them because they developed these so-called nanobots that were specific to the individual's DNA, and so they could selectively target an individual based on their unique features in DNA, and that poison that killed that one individual would only kill that one individual. So to reverse that, that's kind of the reverse of precision medicine. But in precision medicine, we would be able to develop treatments that would work for that one individual perfectly. And so the splitters versus lumpers question. Well, what is splitter and what does lumper mean? So this is a common conversation in anything in science, even apart from medicine. There are people who like to classify everything to the greatest detail, and those are the splitters, where you look at a bird and then you say, well, this is a species. And then somebody says, well, you know what, I want to get my name in the book, so I will just name this subtype of this bird a different name. And then somebody comes and says, well, no, it's just the same bird. So the splitters are the ones who would like to just split and have a never-ending number of classes and subclasses and categories. And lumpers would be the opposite, where we would just put together various subtypes into one and treat them as one. So who is right? Well, I think it depends. It depends on the question, because in some cases, being a splitter gets you to the better results when it comes to practical management. And some other times you got to be a lumper. And I was just discussing it in the intro part that for instance, I'm a splitter when it comes to treating patients in clinic, and I would like to be as individualized and as personalized as possible. If an individual has a certain subtype or has a certain genetic variant or mutation, if that has any practical relevance, I would like to know that, and I would like to individualize the treatment accordingly. And it's especially true when it comes to disease modifying and neuroprotective treatments. In that regard, we need to be as detailed splitters as possible. On the other hand, in some cases, as I mentioned briefly earlier, being a lumper can be practically relevant. If somebody is falling, there may be a good way to have a lumper mentality, because you're not going to have physical therapists who will know every single detail about the subtypes of balance problems, and it has practical value to just train as many of them as possible to have at least some balance therapy skills, that we will make a bigger difference in the community by that. The other way that I am a lumper is I oftentimes use bigger categories when I try to explain things to students and others, to get them started to have an idea about a larger category, and the detail can come later. You know, when you decorate a tree, you have to have a tree, and then you know, the ornaments can come a little bit later. So that's how we stand, and I do believe that the splitter approach will be increasingly favored. We don't know how much artificial intelligence will drive our diagnostics and treatment decisions in the future, but I would approach, I would propose that when there are 200 different subtypes of Parkinson's disease, even the smartest physicians will not be able to formulate and learn and change and know everything in how that would determine individualized treatment plans. So some form of AI will be helpful in how we plan and develop and improve our road towards precision medicine. So I hope that I was able to give the two sides of the argument, or the question of, is Parkinson's disease a single disease? So the answer is yes and no. In some ways it is, and in some other ways it's not. And the part that it's not is far more interesting, because it is hoped to lead us to more effective, more specifically targeting treatments in the future. Thank you very much for your attention, and I think we have time for questions.
Thank you so much. Absolutely, we have some time for questions. And I know one of the very first questions that came up was one that you and I discussed earlier before everybody hopped on. And can you talk about body first versus brain first types of Parkinson's?
So the question is where this whole thing starts, and just like with many things, when we talk about heterogeneity, we talk about how you know, not two people are the same. It comes to that as well, and I mentioned in the discussion before everybody joined that in the current issue of Movement Disorders Journal, there is a very interesting hypothesis that is presented by a group of French authors exactly on that topic. And what they discuss is that it's not the same in everybody. Sometimes it's body first, sometimes it's brain first. And a great example of this gut-brain discussion has been that it's not a simple one-size-fits-all solution. Many years ago, when it was discovered that the synucleinopathy that I mentioned, that is the underlying basis of the brain pathology that causes Parkinson's disease, was observed in the enteric nervous system. When they took biopsies from the gut and they looked at the nerves in the gut, they saw the same changes with this alpha-synuclein that we know to cause Parkinson's disease. So that created a lot of hype, a lot of interest. By the way, the enteric nervous system is huge. There are approximately 10 billions or 5 to 10 billion neurons in the enteric nervous system. It's an autonomous entity. It can control itself, and it does a lot of functions in how the peristalsis and motility of the gastrointestinal tract is controlled on its own. It has some influence from the central nervous system, but very little. So the bottom line is that there are neurons, there are nerve fibers in the gut, and it showed the same changes that are known to cause Parkinson pathology in the brain. And so the next step was a very interesting study that looked at samples taken from colonoscopy of patients who didn't have Parkinson's disease. They were just getting colonoscopy for cancer screening or for other reasons. And then many years later, it was found that some of those individuals who had this synucleinopathy seen in their gut, again, years and years before they had Parkinson's, they didn't have Parkinson's disease at the time those colon biopsies were taken. And it turns out that many of them later, years later, developed Parkinson's disease. And the ones that did had signs of this synucleinopathy in the gut biopsy samples years and years earlier. So that immediately gave rise to this whole hypothesis that Parkinson's starts in the gut. It enters through something that happens in the gut, and then the pathology begins in the enteric nervous system. And then there's one cranial nerve that is the link between the enteric nervous system and the brain. It's called the vagus nerve. And somehow this synucleinopathy that starts in the gut will travel through the vagus nerve into the brainstem, and then it starts the process in the brain, eventually leading to Parkinson's disease. Well, that theory didn't quite work out, because later it was found that, number one, some patients who had that synucleinopathy in the enteric nervous system didn't end up developing Parkinson's disease. They ended up developing another synucleinopathy called MSA, or multiple system atrophy. So the finding of synucleinopathy in the enteric nervous system in these samples was not specifically connecting the synucleinopathy in the enteric nervous system with Parkinson's disease. It could be another disease, so it was not specific. But the bigger problem with the theory was that many patients who do have Parkinson's disease today, and then we look at the enteric nervous system in biopsies, do not have any synucleinopathy in the enteric nervous system, even though they have had Parkinson's disease for 10 years. So the theory just kind of failed, and certainly it did not apply to everyone. And so eventually this paper that I was just mentioning that appeared in the current issue of the Movement Disorders puts it all together, but others have already done similar, these are theories and hypotheses that are mostly unproven, but essentially the idea is that how it starts and where it starts varies from one patient to another. It could be the olfactory bulb. Something enters that we breathe in, and it takes hold in the olfactory bulb. And that's why many patients will have loss of sense of smell, sometimes years before they have Parkinson's disease. But again, not all. And that speaks to how it's not one size fits all. In some patients, whatever agent it is that starts the process comes in through the nose. Some other patients, it comes in with what we eat or drink through the enteric nervous system, and then starts that process. In others, it enters in other ways. One popular discovery was that the same synucleinopathy, which can be proven under the microscope with immunohistochemistry, is seen in the tiny nerve fibers in the skin around the sweat glands. Those are called pseudomotor fibers. And in some practices that I know, they even do skin biopsy to confirm the diagnosis of early Parkinson's disease by looking at those sweat glands in the skin biopsy and looking for that synucleinopathy there. Well, first of all, if this is something that is offered to you, I don't recommend it. It doesn't really have practical value. I mentioned earlier that Parkinson's disease is a clinical diagnosis. Any movement disorder specialist can do that without skin biopsy. So, and it's not FDA approved for that purpose either. But the bottom line is that a lot of patients will have those changes in the skin biopsy in the tiny sweat gland nerve fibers, but others don't. And why is that? Well, that's because in many patients, it doesn't start in the skin. It doesn't get into the skin. In others, it does. And then finally, there are patients who will have no sign of synucleinopathy in the olfactory bulb, in the skin biopsy, or the gut, none of that, but they will have it in the brain. So how did it get into the brain? Well, we believe that it can just start spontaneously in the brain. And I mentioned those risk genes. There is a mutant alpha-synuclein so-called H53 gene where the synuclein gene itself is mutated. Now, those individuals, almost all of them, will have Parkinson's disease, regardless of what environment they are exposed or not exposed. And in fact, if you put that mutant synuclein gene into mice, the mice will have synucleinopathy and Parkinson-like pathology in their brain, every one of them. And so what I'm trying to say is that on its own, this particular protein called alpha-synuclein can start becoming bad without any infective agent or any other influence or environmental exposure, maybe in a small percentage of patients on its own. So the bottom line is that brain versus body first. The answer is in some patients, it's in probably more patients, it's body first. The process enters through one of those entry points that I mentioned, and then it makes its way to the brain. But it could be the brain first. And in fact, a research group recently demonstrated that there is such a thing as brain to the gut. So I mentioned that years ago, there was this big enthusiasm about the gut-brain connection, where the synucleinopathy starts in the gut and makes its way to the brain through the vagus nerve. And then later it was refuted as at least not universally present. And now it has been demonstrated that the opposite can happen in some patients too, where it starts in the brain and through the vagus nerve reaches down to the enteric nervous system and causes the synucleinopathy there secondarily to the brain. So again, the question was brain or body first. It could be either. It varies. In certain patients, it's body first, probably more patients like that. And in certain patients, it's brain first.
And just for clarification, someone wanted to know that you mentioned at the beginning that people with Parkinson's have Lewy bodies. So can you just clarify again the difference between Lewy body and alpha-synuclein?
So the bad alpha-synuclein makes up the Lewy body, because what happens is that once the alpha-synuclein, which we all have, becomes bad and toxic, it comes with it becoming water insoluble. So inside the cells, inside the neurons, there's a sort of a liquid that's called cytosol, which is a collagen liquid. It's not like water flowing freely, but it's still a water solution for the most part, with all sorts of little structures that organize the workings of the inner cell, enzymes, structural proteins, etc. Now, in order for anything to not aggregate into inclusions and little bodies in there, they need to be water soluble. And if something is not water soluble, then it starts aggregating. And so that's what happens when the alpha-synuclein has a conformation change. It goes from water soluble to water insoluble. And that's when it starts making these clumps. And when the clumps get big enough, then you can see it under the microscope. And that's what makes up the Lewy body, which is a pathological hallmark of Parkinson's disease. But you can see Lewy body in Lewy body dementia as well. And the whole process is extremely similar at the cellular and molecular level. In fact, it's indistinguishable between Parkinson's disease and Lewy body dementia. What distinguishes these two conditions is not what happens at the level of the cell, because that's the same. You see Lewy bodies, just like I showed you on the slides. You see the picture at that level is identical. What's different between Lewy body dementia and Parkinson's disease is the proportion, how this spreads, how fast it spreads through the brain, because this process propagates. This bad synuclein, when it comes in contact with the good one, then just like prion, some people might have heard about prion diseases. When the bad protein gets in touch with the good one becomes bad one, and the whole thing just goes on to the next cell and the next structure and the next part of the brain. Now, in Lewy body dementia, that process is faster. It goes faster. And number two, the different parts of the brain are not created equal as far as their resilience, their ability to withstand the pathology, to survive, their sturdiness. And it turns out that different brain areas will succumb to this process at different times and different extent. And what's different about Lewy body dementia compared to Parkinson's disease is that the higher order part of the brain, called the cortex, gets affected, involved, and brought down by this pathological process earlier, quicker, and more severely. And that explains why it clinically is a different disease.
And in regards to risk genes, there was a question: if someone does have a genetic predisposition, we know you've mentioned certain triggers like environmental triggers. Can other triggers like stress or chemotherapy or any other things like similar to that, anesthesia, trigger Parkinson's?
We don't know that, but it's very likely that many factors, such as the ones you mentioned, will play a role in some way or another. Maybe not at the molecular level, but at the system level. You know, I always say that we need to talk about comorbidities more. Because if somebody has, let's say, white matter disease from cerebrovascular risk factors, or have another process, aging, the age itself is a factor, then they don't have as much of a reserve, and they don't have as much of an ability to use neuronal plasticity to compensate for the Parkinson pathology. So that alters the whole process. And so you could have the same genes or genetic predisposition, but then if you have cerebrovascular disease, or you have traumatic brain injury, or you have a tumor, or you have, as you say, chemotherapy that kills some of the neurons, all of these things will substantially alter the process, and you would look like very different night and day, even though your genetic predisposition, where you come from, is the same. But these factors can completely derail the pathological process in clinically very relevant ways. Now, how we break it down, which one, it's not a single one. It's certainly a multifactorial system. And just like things can make the process faster and worse, there may be some protective factors too. Exercise, what you eat. You could have a bunch of protective genes that that may, your parents lived to 100, and you have a naturally and genetically higher potency in neuronal plasticity potential, which can compensate for the decay of the tissues from the Parkinson disease. You will have a less progressive and less severe disease because of those other factors. So there are, sort of factors on both sides of the aisle.
Awesome. Well, this has been very enlightening because it's a very complex topic, and you've done such a great job really breaking it down for us. So I want to thank you so much about covering all of this so well. And I know everyone with this is very much appreciative of that. So know that we're out of time, and I know we didn't get to all of the questions, but I think you covered a lot of these questions throughout your conversation today. So I want to thank you. And we have a tradition here at PMD Alliance as our thank you. It's our wave of gratitude. So if you guys are on screen and are willing to give the wave of gratitude to Dr. Mari, we thank you so much for this great talk and for everyone joining us. It is being recorded, so it should be available on the website within a day or so. We will have slides available that will be attached with that. So you'll be able to go back, because I know it was complex. I know we covered a lot, so you can go back and review it and, take your notes, because I know I've been taking notes and learning a lot. So thank you so much, Dr. Mari.
Thank you for inviting me, and thank you for the questions and the attention. It was my pleasure.
All right. And thank you everyone for joining us, and we'll see you soon. Take care.
Okay, bye-bye.
Bye-bye.